dysfunction in the organs, means disease. So in his disease, what functions are affected? So first you have to know the functioning, normal functioning of the body, of the organs, of the organ systems, of the tissues, of the cells. Unless you know the normal functions, how can you study the dysfunction? Isn't it? So it is the foundation. In medical practice, blood, this chapter, hematology is the foundation. You must know about this normalcy. So you have to concentrate, you have to learn thoroughly, and every time it will be required by you while you will be in practice. So it is very important everywhere, not only this, in your exams, these are also very important. In practicals, you will be having a lot of practicals regarding this blood. So you have to perform practicals. Okay, and today itself we will be starting practicals. And the first practical will be microscopy. I know a lot of you are not able to focus microscope properly. So we will be learning in 2 sessions how to focus a microscope, what are the different parts of the microscope, how these are handled. Focusing, light, how much light it will be entering into the microscope, depend on that how the things are seen under the microscope, it will depend. More the light, less will be the visibility. Less the light, less will be the visibility. The resolution of the image will be not proper. So you have to learn this. Moreover, today I am taking a class, taking on this. Immediately after lunch break, you will be going to, one group will be going to the physiology department, another group will be going to the biochemistry, another group will be going to anatomy. The groups who will be coming to physiology department, they will be segregated into 4 groups. Okay, and we will make some small groups. And with these small groups, again we will be discussing this with you, and you have to interact with us. Here it is not possible to interact with all of the students, but there you will get the opportunity of interacting. So same thing, here you will the basic thing, here more elaboration will be there. Your doubt can be cleared over there. So that is important. Okay? In the coming Saturday, there will be a very important class. I told you, Wednesday and Saturday, I will be taking the class. Saturday, there will be a class on plasma proteins. Okay? After that, there will be one class, ECE, Early Clinical Exposure. So what we will be doing there? We will give you some problems, cases. Those who are having some blood related problems, you have to identify what is the problem, what happened to the person, and what is the explanation. Lot of questions we will be asking, whether you will be able to answer those questions. This is the EC, and it will continue for 3 hours. Here I will be conducting it. Okay? It will be related to this topic and related to plasma protein topics. So you have to learn this topic. If you do better in the ECA classes, here you will be also assessed. And there is another class, SDL, self-directed learning. In the self-directed learning, the last 1 hour, you will get questions through Google Form. Some questions you will be facing. And within this 1 hour, you will MCQ. These are all MCQs. So you have to tick which one is correct. Only one scope you will get. Once you tick it, it cannot be changed. Automatically, through online, this will be coming to me or to the assessment section. Okay, so there will be 20 marks, 20 MCQs. So if you learn this, and these are component of internal assessment. Okay, so take it seriously. Understood? Okay. One more thing. When I entered, definitely you don't know whether I am a teacher or not. Since you have entered, I think attended yesterday's program, might have seen me. So you know that I am a teacher. When a teacher enters, what is your duty? To wish him but I didn't follow up. Some 3 or 4 students, they used to do this when others were remain seated in the, so ignored me. So not only, you can ignore me, okay, I am a bad teacher, so you can ignore me. So you should give respect not only to your teachers, to your seniors also. The same manner. You have to wish them, you have to give respect to them. Otherwise, I will get the help from them. So this is requirement. Okay? Fine. And in physiology, we have 12 areas, 12 topics. The first topic is general physiology. Dr. Balaram, he will be addressing this topic. His classes will be on Monday and Friday. I am taking on Wednesday and Saturday. So weekly you are having 4 physiology classes. So first topic is general physiology, and the second topic is this one. See, competency number 2.1, it means topic number 2 and subtopic 1. Okay, so Balaram sir, he will take 1.1, means general physiology. And so general physiology, blood. Then NM physiology means neuromuscular physiology. Okay, then comes respiratory physiology, all the organ systems will come. In this way, there will be 12 topics. And the last topic is integration. How all the topics are integrated. So you can expect questions from all these topics. CVS, cardiovascular system. There will be 6 topics in first paper and 6 topics will be in second paper. And what is the type of questions that I will discuss, that I will just let you know so that you can prepare yourself. How in either it is first paper or second paper. What is the form? The first, there is what? MCQs. You will be given 10 MCQs. You have to finish it within first 15 minutes. After that, answers will be taken back. MC? Okay, so you have to, and the marks for this is 10 marks. Out of 100, 10 marks gone for MCQ. Then there are 90 marks. Okay, though now the second question is structured essay type questions. Structured essay type questions means there are 10 marks you are having, 10 marks you are having, and this is structured, means it can be fragmented. Means maximum we can allot 5 marks, 4 marks, 5 marks, at the most 6 marks. Then remaining marks will be 2 plus 3 this way. So this is structured. Okay. Say for example, describe the intrinsic pathway of blood clotting, coagulation. So it may carry 5 marks. Remaining marks will be related to this topic. Okay, this way you can expect 10 marks. And the third question is short notes, and these are all applied aspect, disease related. For example, in blood, we will be studying anemia. So short notes, write short notes on anemia. We will be studying jaundice. So write short notes on jaundice. Okay? Myasthenia gravis. Okay? So these are the applied aspects. So there will be 20 marks. And there will be 5 questions. You have to answer 4 questions. 4, 5, 20. I am giving you this idea because you will be able to prepare yourself what type of questions will come and how much you have to write. Number of marks is also, you can, in this type of short notes, there are how much mark? 5 marks. How many answers you have to do? 4 questions. Next comes explanatory type of questions. Here 3 marks are allotted. What is this? Very interesting, these questions. For example, blood while flowing through the vessels, circulating blood, does it clot? Does it clot? No. But whenever you take out, it clots. Why? Explain. I will explain it today. So these sort of questions may come. Plasma protein deficiency may lead to edema. Explain. Myasthenia gravis is a neuromuscular disorder. Explain what happens in myasthenia gravis. So these are severe. The wall of the heart is made up of different layers. Hello, the last benches? Are you listening properly? Okay. If you find difficulty, you can come here. Okay. Inner layer is most prone to MI, myocardial infarction. Explain. Every organ gets more blood during systole of the blood. But our wall of the heart, made up of myocardium, these muscles get less supply during systole. They get more supply during diastole. How come? Explain. So these sort of questions you may expect. While I will be studying all the topics of the subtopic, I let you know. So you may face this sort of question in your exam. So accordingly, you have to prepare those questions. Understood? Fine. Next, second half. Second half means first half, second half. The question, these first half questions you will be answering in an answer book. For second half, you will be having a second answer book. Second half questions, there will be separate answer book. So you have to answer all this in the second. Okay. There will be, again, you will be getting short answer type questions, and all will be having 5 marks. You have to answer 5 questions, such questions. Okay. And you will be given 6 or 7 questions. Out of that, you have to answer 5 questions. Okay. Then short note is also there. Then one short note or question will be from ATCOM. What is ATCOM? A stands for ethics in medical professionalism. In your profession, what should be your ethics? And ET for ethics, communication. How you should communicate with your patient. What is your responsibility to the society? What is your responsibility to the patient? what the patient expects from you, how you should deal, how you should communicate with the patient. So all these things are taught in AdCom. So there will be questions. AdCom syllabus is there separately for anatomy, separately for physiology, separately for biochem. Okay, so in all subjects, one AdCom question of 5 marks is compulsory. So you attend AdCom classes. And this AdCom will be continued up to your final phase. Second phase, third phase, every phase there will be AdCom. Okay? So you got the pattern of questions. What would be the pattern of questions? In blood, what are the competencies? There will be, these are the competencies. So this is about blood and this is about plasma protein. This is about RBC hemoglobin. This is erythropoiesis. Hemoglobin is variations, significance result. Here it will be polycythemia and some applied aspects, jaundice, anemia, everything will be discussed here. And it is about WBCs, different types of WBC, their variation in different diseases could very important this part. Normal amount of WBCs is this much. Under what condition its level is increased, under what condition it is decreased. You have to know that. Okay, so these are the applied aspects we will be knowing. Then we will be knowing about immunity. We know there is innate immunity, different types of immunity, so we will be discussing immunity. And the platelets, thrombocytes, describe the composition, function, pathways, mechanism of action of anticoagulants. Here the blood-related disorder, hemophilia, purpura, applied aspects. So you will be learning this. And this is all about practicals. You will be doing estimation, you will be estimating hemoglobin, you will be counting how many RBCs you are having in your blood. TSC means total leukocyte count, and DLC, differential leukocyte count. You have to perform all this. And you have to determine blood groups, you have to determine bleeding time, clotting time, RBC, different RBC indices are there, like hematocrit, packed cell volume. All these experiments you have to do in the practical classes. Okay, so this is one. Then reticulocyte, immature blood cells, right blood cells. So you have to study about the reticulocyte. You have to study about the ESR. Might have, but you don't have to perform this. But you have to know what is ESR, under what condition these are varied, in what pathological condition, in what physiological condition it shows variations. So these are important for your biopsy, as well as you won't be expecting any long type of questions from here, but for biopsy exam, biopsy is very difficult. A lot of marks is there for biopsy. A lot of marks. Okay, external examiners will cross you. So prepare, if you want to get proper marks, so you have to prepare yourself. You have to be very sound about this. Okay. So today, as I told, I will be elaborating most fundamental and fascinating topics in physiology, that is blood. It is called river of life. What does it mean, river of life? This river, through this river, what flows? A red fluid. Why it flows? It flows to every cells, almost every cells, every tissues, every organs of the body. Why? What is the need? What is the necessity? Necessity is the cell for its survival, for its life. It gets everything from the blood. Whatever it produces, it gives to the blood. And it is taken away. Who will be needing this? It will be supplied to them. Or if it is not necessary, that it will be excreted. So blood is supplying all the necessary things to the tissues, to the cells, every cells. That's why it is important. That's why it is called life. you can survive without food for some days, isn't it? You can survive some days without water. But without blood, without oxygen, your brain tissue may survive for only within 3 to 10 seconds without oxygen. If you stop the supply of oxygen to your brain, within 3 to 10 seconds you will be fainted. You will be unconscious. And if it continues, there will be permanent damage of the brain cell. We call it stroke. Cerebral thrombosis, or different reason, there is cerebrovascular accident, CVA. Okay, so it may happen. So as I said, blood touches every tissues, every cells. Why? Because blood performs various functions coming. What are the functions? Lot of functions that are performed. You have to remember all those functions. Okay. And you see, very interesting thing. You will be knowing the normal composition of blood. Okay. So these are the normal composition. This component, normal range is this. But this range is changeable. It may be lower than this, it may be higher. And it happens during diseased condition. Okay? If my organs shows dysfunction, it will be reflected in blood. It will be reflected in blood. So any dysfunctions of the organ, this is reflected in blood. Okay, that's why we study blood. Whether any dysfunction is there in the body system. At the same time, if any components of the blood becomes higher or becomes lower, then it may affect the organ. Again, reversely, it can affect the organ system. Understood? Disease will affect blood composition, and changes in blood composition may affect your organ systems. For example, acidosis. Blood becomes acidic. It may affect your brain, it may affect your heart. Red blood cell, if you produce too much of RBCs, it will affect your body. Similarly, if you produce less hemoglobin, you will become. See, one of the case came to you. One student, suppose, one student who were performing experiments in physiology lab. Suddenly, she became fainted, unconscious. Okay? And immediately, when you observe, her pulse rate is very fast. And blood pressure is low, below normal. Immediately, we tested her blood and it is found hemoglobin concentration is 6 grams per cent. What happened to her? This is the easy question. What happened to her? Can you tell me? Anybody? Some changes is there in the blood. And because of this, she suffered, her cardiovascular system is not functioning properly. Heart rate is fast and blood pressure is low. She became fainted. This adequate blood is not coming to the brain. Faint means adequate oxygen is not supplied to the brain. What happened? She is anemic. She is anemia. She is actually suffering from anemia, unable to supply required amount of oxygen. If she is unable to supply adequate nutrients to the cells, cells cannot generate sufficient energy. She becomes lazy, she becomes fatigued, she becomes weak. Okay, so we can see only lowering of hemoglobin can affect body. That's why studying blood is very important. Okay, even as I explained, for exam purpose it is also important. And what is hematology? What is hematology? The topic is hematology. What is written there? The study of physiology and blood disorders. Okay? So, for this, what you do? Study of physiology of blood. Physiology means what? All the components. What are the whether their amount components are adequate, whether say RBC, whether number of RBCs are adequate. Okay, RBC is produced, but they may not be normal. For example, in polycythemia vera, the WBCs may not be normal. For example, in leukemia. So, whether their appearance, morphology, functions are normal or not, that we will be studying here in physiology. We will be studying different disorders of blood. And what is the etiology? What is the pathophysiology? What are the reasons? That are we study in hematology. Okay? So these are the lot of things will be our learning objectives. Everything we have to learn. Lot of objectives we are having. I will not confine on this. Everything you will be learning. Now blood. What is blood? Who can tell me what is blood? Very simple. Blood is a red colored connective tissue that flows through the blood vessels and supplies a lot of things, performs various functional vessels. These are liquid connective tissue, isn't it? Why you call it connective tissue? Connective tissues have some characteristics. What are these characteristics? They might have matrix. They must have matrix. And within this matrix, the cells, fibers are embedded. They are embedded. They are suspended. In our blood, we also get matrix. Can you tell me what is the matrix? Plasma. Liquid matrix. In which cells are suspended. Not only this, they connect different parts of my body, different tissues, different cells of the body. That's why we call it liquid connective tissue. I may ask you why the blood is called liquid connective tissue. I think everybody realized, understood why it is called liquid connective tissue. Okay. And they are derived from, one of the characteristics is they are derived from mesoderm in vascular system, and this blood is pumped by heart. Let us study some properties of blood. Very important. Properties of, I will come one by one in detail. Forget this slide. First is color of the blood. Who can tell me what is the color of the blood? Sir, red. Then how can you differentiate blood of, by seeing the blood of arteries and blood of vein? The color, intensity of the color. The arterial blood will be bright red and the venous blood will be dark red. Why? What is the reason? Arterial bloods are oxygenated, mostly oxygenated blood. Hemoglobins are oxygenated and they produce bright red color. And venous blood are deoxygenated, means there is more carbon dioxide the hemoglobin is carrying, That's why its color is dark red. So this way you can differentiate. Now, there are some disease conditions, the color of the blood can be changed. For example, in purpura, if somebody is suffering, what happens? If you become afraid, so much afraid, you lost control. You become blue, isn't it? Why you become? Because of cyanosis. Means you are unable to take normal oxygen and carbon dioxide is more, carbon monoxide, carbon dioxide and hemoglobin, hemoglobin is produced. More hemoglobin is produced and this gives this cyanosis color, deep blue coloration in the skin. Okay. In cyanosis, bluish discoloration of skin due to reduced hemoglobin. Also, you can get the cherry red color of blood in what condition? In carbon monoxide poisoning. Cherry red color you will get in carbon monoxide poisoning. Remember this point. And methemoglobin it gives brownish blood. What is methemoglobin? Hemoglobin, we will study. Hemoglobin has different variations. One is methemoglobin, it is the abnormal hemoglobin. Normally, hemoglobin contains an element. Can you tell me the element? Iron. And it remains in what form? Remember this point. In what form? Ferrous or ferric? Ferrous form. If by any means in your bone marrow the hemoglobin contains ferric form, then it is called methemoglobin. It is abnormal hemoglobin. Okay, so it is seen in methemoglobinemia. Emia means blood. Methemoglobin in blood. Clear? Okay. This is about color. And the volume. Important. You might be facing the questions. What is the normal blood volume in a adult female? What is the normal blood volume in a normal adult? 5 to 6 liters. 4 to 5 liters. So that is the range. Average we have 5 liters of blood. There will be 4.5 liters of blood. Average 4.5 liters. Average. Also, how much blood you can have, you are having. Suppose your body weight is 60. In male, it is 7 to 8%. Suppose it is 8%, then 6 into 8, 4.8 liter. So you will be having 4. You understood this? That is 8% of your total body weight. In female, it is 7%. There is it, 6 to 7% of your total body weight. For male, it is 7 to 8%. Remember this. You may face MCQs on this. Who knows? It may be there in SDL MCQs. So you learn this. And adequate blood volume is required. Very important. Why? For proper perfusion of your tissues. If it is underperfused, we use the term hypovolemia. Remember this term, hypovolemia. Hypo means less, volume means volume, emia means blood volume is less. Less blood will be going to tissues. Say in shock. What is shock? You will be facing this term, shock. What is shock? Every tissues, brain, including brain, should have a minimum this much blood supply, blood flow to the organs. If it is below this normal, then the condition we call shock. The patient faints, become unconscious because of shock, because of less flow of blood into the organs, into the tissue. Clear? Okay? So, hypovolemia is seen in shock. It is shock seen in dehydration. You are not drinking any water for the whole day, but working in sunny environment. What will happen? You will be losing water from your body through evaporation, conduction, convection. So various processes you will be losing fluid. Which fluid are going out? Tissue fluids. Now if the tissue fluids comes out, then whether the tissue will be working properly? No. So that's why we have a regulating mechanism. Our emergency condition, tissues will not work. But our plasma is having 91% water. So from this water, I may supply some water to the tissues. So some water from the blood will be migrated to the tissue fluid to save the tissues. As a result, what will happen to the blood? Blood will become more concentrated, less water, more cells. Isn't it? This is we'll call it dehydration. In this condition, blood level will be less because water is shifting to the tissue fluid. Okay? Next, hypervolemia. Means blood is more in your body. This is called hypervolemia, more than the normal amount. Hypovolemia means less than, less than 4 liters for you. Okay, for them it is less than 3.5 liters. So this is hypovolemia. And hypervolemia in heart failure, in edema. I will explain edema. How edema, when I will start explaining you the plasma proteins and their role in capillary perfusion. So in that you will be learning. See, you will find small variation of plasma protein can create a havoc. Okay, so learn this. There are stalling process. What is question comes? What is the stalling process? And how they are involved in tissue perfusion. Reactions of blood means pH. So pH, our pH range is 7.35 and 7.45. If the pH becomes below 7.35, the condition is called what? Alkalosis. Sorry, acidosis. Below this, going towards acidity. Over this, alkaline. So when blood pH becomes more than 7.45, the condition is called alkalosis. When alkalosis is seen, it is seen in hyperventilation. If you hyperventilate, continue for some time, there will be alkalosis. Lot of carbon dioxide will wash out. At the same time, there will be deprivation of bicarbonate ion in the blood. As a result, your blood will become alkaline. Okay. So in vomiting, your lot of bicarbonate ions are, vomiting means, diarrhea means, your water will be coming out, and through this water, a lot of bicarbonates will be going out. As a result, you will be suffering from alkalosis. And in diabetes, there is ketoacidosis. I will explain. Ketoacidosis in renal failure. This acidosis is a serious problem. In the patient may die, may undergo coma. If it is too much acidic, patient may undergo diabetic coma. You might have heard diabetic coma. So it is very important to maintain the blood pH within this normal range. Any deviation, there will be pathological change in your body. And specific gravity. Blood is the specific gravity of blood is 1.50 to 1.60. In plasma, it is 1.20 to 1.30. 1.0 20. So its significance is specific gravity. It depends on number of molecules. So if the number of molecules is increased in the blood, specific gravity will be increased. And the specific gravity is because of increase in plasma protein concentration. When this plasma protein concentration can increase, one is dehydration. Dehydration, blood is lost, blood is become constant, this plasma protein level is increased per 100 ml. Okay, so this condition may lead to increased specific gravity. And decreased specific gravity in blood, your number of cells will be reduced, so specific gravity will be reduced. Less specific gravity, blood flow will be fast. More specific gravity, blood flow will be less. That means blood specificity is resistance to blood flow. Viscosity. What is viscosity? Viscosity, in terms of, it is 4 to 5 times heavier than water. 4 to 5 times, remember this, 4 to 5 times heavier than water. Mainly due to plasma proteins and hematocrit. Here I have placed here one term, hematocrit. We will explain that now. What is hematocrit? And significance, peripheral resistance and blood pressure is dependent on viscosity. Higher the viscosity means more the blood viscous, more the resistance will be there in flowing blood. Blood will face more resistance while flowing the blood vessels. So more resistance, increased blood pressure. So increased blood pressure, lot of problem. Okay, so viscosity is important. Okay, in dehydration, viscosity is increased. If you take lot of water, viscosity will be reduced. So reduced viscosity will cause less vascular resistance. Blood will flow easily. Temperature. Why we maintain our body temperature constant? Why? We maintain our body temperature constant, isn't it? When the temperature goes up, we have been suffering fever. When temperature goes down, below normal, hypothermia. We call it hypothermia. What happens? In fever, infections or inflammation, there is increased blood temperature. And when the blood temperature is increased, okay, why this temperature we have to maintain? Because you have learned all the cellular functions are governed by some enzymes. And every enzyme has isoelectric pH. Okay, and another is temperature is a fixed temperature. At this temperature, enzyme activity is maximum. If you increase the temperature, there will fall of activity. If you decrease the temperature, optimum temperature. Say if the optimum temperature is increased, initially rate of metabolism will be increased, then it will fall because the proteins, means enzyme, will be denatured. Okay, if you decrease it, again metabolic activity will be, enzyme activity will be decreased. That's why we try to maintain temperature constant. Okay, so this is the importance of osmotic. And then comes the osmotic pressure. Blood has provides a total of 5600 millimeter of mercury is the total osmotic pressure. But for us, the plasma oncotic pressure means plasma protein, the plasma proteins, they exert their colloids. Plasma proteins are colloids, macromolecules. They cannot pass through semi-permeable membrane. Okay, so by remaining in the blood bag, they exert a pressure, we call it oncotic pressure or osmotic pressure. And this is 25 millimeter of mercury. And because of this, are you having edema? Swollen tissue? No. Where? Because your osmotic pressure is normal. When your osmotic pressure will be reduced, your body will be swollen. Taste of, we'll discuss this point. Taste, is salty because of lot of sodium chloride, sodium ion is there, sodium chloride. Why? What is the need of sodium ion, sodium chloride in blood? all the cells in our body is living. Okay? And they are having a membrane, and this membrane are having a resting membrane potential. And this resting membrane potential gives you, in fact, these cells are excitable and they are living. And this is maintained by sodium ion. Not only sodium, there are different ions. Sodium ion is one of the important. Their concentration is higher outside the cell, concentration is less inside the cell. By some other factors, they maintain the resting membrane potential. And those who are having resting membrane potential, they can be excited for my muscles, from nerves, they can be excited. But those who are not having resting, they cannot be excited. Okay, so this is important for excitability, and for excitability sodium ion is very important. That's why we need sodium. Excess sodium ion can be accidental, can be hazardous. Hypernatremia. It is called hypernatremia. Less sodium in hyponatremia. These have chronic consequences, hazardous consequences. Okay. Now the functions of blood. What are the functions of blood? Functions of number one, respiratory functions. While I will be discussing the function separately in another class, RBC, in that, since I am having very less time, so respiratory functions, we have transport meat. It is a transport medium. Through blood, various things are transported, like nutrients, like excretory substance, urea, uric acid, creatinine, xanthine, hypoxanthine. These are excretory products. They are from tissues, they are taken through the blood to the excretory organs, kidneys. They are from where they are eliminated. Okay, so excretory functions are performing. Water hemostasis, I already told. We have to maintain the water. When there is dehydration, there will be movement of blood from plasma into the tissue space. When blood is having less water, the blood will be coming, water will be coming from tissue space into in this way, interchange will be to maintain the hemostasis. Temperature regulation, storage, body color, oncodic pressure. In details, everything will be detailed out when we will be coming to specifically for those functions. Before I go into composition blood, I want to elaborate some concept I want to give you. Then we will enter into composition of blood. What are these concepts? One is coagulation and clot. What is coagulation? Blood to fluid. It is a mechanism, protective mechanism, by which this fluid is converted into solid, semi-solid, jelly-like structure. The mechanism by which it is formed is called coagulation or blood. What is coagulation? The mechanism, the protective mechanism by which the fluid, blood, is converted into semi-solid, jelly-like structure. This is coagulation. Okay? And there are various processes involved with this. We will study. And these are very important for you. Long type of questions, short type of questions, all these are important. So you have to learn it carefully. So coagulation and the clot, the semi-solid, the structure we got, it is called clot. Okay, so it happens, and why it happens? I told it is a protective mechanism. That means what protection? It prevents hemorrhage. It prevents loss of blood. Think, if the mechanism is there, when a blood vessel is cut, then what will happen? Continuously you will be losing blood and you will die. But this is prevented by this mechanism. That's why it is a preventive mechanism. And there are such substances, they can prevent this mechanism. And these are called anticoagulants. What are the names? A few names. What is this? Sodium citrate, sodium fluoride, EDTA. Bible question. What is the full form? What is EDTA? Write it down. This is EDTA. What is ethylene diamine tetraacetate? Even today, our tutors may ask you, what is EDTA? So EDTA, it is an anticoagulant. The full form is this, heparin. Is another anticoagulant. Okay? What is thrombus? What is thrombus? You might have heard this word, thrombus and embolus. Blood can be coagulated within the blood vessels because of problem in the endothelial lining. For example, atherosclerosis, blood inner lining becomes irregular. When blood flows through this area and comes in contact with the atherosclerotic plaque, then what? Platelets become activated and starts coagulation. So a clot is formed. Intravascular coagulation is the clot formed within the blood vessel is called thrombus. A part of thrombus can come out and enter in the smaller vessels, ultimately block a vessel by this small part. This small part is called embolus. Multiple fragments, emboli. So you understood what is embolus, where is clot, where is thrombus? So these are the things. Circulating blood doesn't clot. Why? There are three important reasons for the, remember all those points. Today I will ask in small group discussions. What is this? The inner lining of blood vessel is smooth. And this smooth lining prevents activation of platelets, addition of platelets, aggregation of platelets, which are needed for blood coagulation. So they prevent it. And they also secrete something. This endothelial lining, they secrete prostacyclin and nitric oxide. Because of this, they can prevent intravascular blood flow. It is the role of endothelium. Okay, smooth lining, one thing. Second, they release nitric oxide and prostacyclin. This is one point. Second point, presence of some anticoagulants within the blood. What are these? These are heparin, antithrombin 3, protein S and protein C. These anticoagulants are normally present within the blood vessel. What are these? What are these? Remember this thing. Okay. And third point is blood is flowing continuously. Suppose one blood factor become activated. Okay. Since blood is flowing, this activated factor will be diluted and ultimately washed out. So, for these reasons, blood doesn't clot inside the blood vessel. Can you remember all these points? Sure? Okay, fine. Next class, I am taking out the blood into a syringe, into a test tube. What happens? What happens? Blood clots. It was in the blood vessel, no clot, but as soon as I take it out from the circulation into test tube or in the syringe, it clots. What happened? Smooth line is absent. Endothelial lining is absent. And second, prostacyclin, nitric oxide is absent. Thromboplastin, thrombomodulin, these are absent. These are properties of this protection is not there. And some blood factors, coagulation factor, they were inactive in the blood vessel while flowing. But when they became calm, they come into the contact with the wall of the glass, wall of the syringe, wall of the test tube, they become activated and coagulation starts. As a result, clot is formed. And thirdly, stasis. Here, since blood is not circulating, the activated factors are not washed away, they are not diluted because it is stasis condition. So they gradually become activated and clot is formed. So this is why when blood is drawn out of circulation, it clots because the natural protective mechanism inside vessels are lost. So clear? So now you can explain these two conditions. What is this? Centrifugation. We are entering into composition of blood. Centrifuged. Blood is taken out in a test tube, anticoagulant is added, then it is centrifuged for half an hour. So then what we at 2000 to 3000 RPM, the revolutions per minute, then take out the test tube, you will get this test tube, this one, you will get two distinct layers, upper liquid layer and at the bottom there will be cellular layer. And this layer is made up of RBCs and these are plasma. In between there is a buffy coat and this buffy coat contains WBCs, platelets, abnormal cells. For example, somebody is suffering cancer, abnormal cells will be there. So these abnormal cells will be present here. Some parasite has infected the blood cells, they will be present here. So this layer is very important. Pathologically, these are very important. And the bottom layer, if you take 100 part of blood, 100 ml of blood, normally this will be 45 ml and this will be 55 ml. And 1% is, out of 100, 45 ml is blood cells and this is called packed cell volume. Percentage amount of cells in blood, it is 45%. And percentage amount of plasma, 55%. And what is the percentage amount of buffy coat? 1%. If you are anemia, if you are having anemia, what will happen to this coat? What will happen to this? Whether it will be less or it will be more. Less RBCs formed. So this experiment, this is called PCB or hematocrit. By knowing the hematocrit or PCB, we can tell whether somebody is suffering from anemia or polycythemia. Increased amounts is polycythemia. Okay? Okay, thank you. See you on